Hey, honors biology people. This is Mr. Coleman, and I wanted to give you a quick little video lecture on a section from your textbook, actually, section 11.4 in the evolution section, about what is called the Hardy-Weinberg equilibrium. Hardy and Weinberg were two separate scientists back in 1908. They actually independently put forth this idea. Hardy was a British mathematician, and Weinberg was a German physician. And what they did is they created a mathematical model that gives us some understanding into how populations evolve. I always like to think of it like they actually, in trying to sort of disprove evolution, they actually prove that evolution occurs. And it's kind of a weird way to think about it, and I'll explain why here in a little bit. But the basic idea here is that this equilibrium will give us a framework of how to understand how populations evolve. So the way that it works is, like I said, by disproving evolution, you actually prove it. In other words, this equation is a mathematical model that shows that populations do not evolve. And they do that by examining specific populations' gene pool. And that's sort of, it's a real mathematical sense, but it does show very clearly that evolution does not occur from generation to generation under certain circumstances, and that is the key. And so here's the gist of it right here. This isn't the mathematical part, but this is where you got to think about it. The way that Hardy and Weinberg's equilibrium works is it describes populations that are not evolving. In other words, if I was to calculate out a gene pool for, let's say, these penguins, and I calculate it out for one generation, and I get 60% of the gene pool is a dominant allele and 40% is a recessive allele. Then I have them go through a generation of mating. I take the F1 generation and I do the same thing. And those numbers should be the same, 60 and 40. And then I take the F2 and the F3 and the F4, and I can do as many generations as I have access to. And what the Hardy-Weinberg equilibrium says is that those populations will not change or evolve because the gene pool percentages are not changing. But what it does say is that in the real world, there are typically 1, 2, 3, 4, or 5 conditions that are happening that sort of cause evolution to occur. So for Hardy-Weinberg equilibrium, here are the conditions that must be met. You must have a very large population with no genetic drift. We'll discuss exactly what genetic drift is, but it's basically animals moving, which goes along with the next statement. No emigration or immigration. So there's no gene flow. So animals can't move between populations. There cannot be any mutations at all. So no new mutations will occur. Mating has to be completely random. There is no sexual selection. And there can also not be any natural selection. So all the traits, they aid equally in the survival rates. There is no survival of the fittest, if you will. So that's what has to be met for this equation to work. Now you can kind of see there's some, obviously, things there that aren't quite right. But let's take a look at the actual mathematics of it. What we're going to try to do is, before we get to mathematics, I do have right here, this slide kind of just explains what we were just saying. Real populations rarely meet all 5 of those conditions. That being said, sometimes for a very short period of time, it's possible. It's possible that for generation upon generation, there may not be any evolution, but over a big span of time, that deep time we were talking about, there at least one, if not all 5 of those conditions are going to happen. So again, it's a model. It's a computer or a data model versus an actual model, and that's how we're going to take a look at it. So this should look familiar. This is the basis of the Hardy-Weinberg equilibrium. What they've done, or what they did, is they were trying to use basic genetics and Punnett squares to predict the frequencies of the alleles. So basically the percentage of the alleles that occur in populations. And you can see there from that Punnett square, it looks a little different. Instead of having a dominant and a recessive letter, there's a P and a Q. If you look at the variables on the side here, the P right here, the P represents the frequency of the dominant allele. So in this case, if you look at it's about fish, the dominant will be the P, the recessive will be the Q, and then the P squared and 2PQ and Q squared are how you would actually figure out how many fish, not the frequency of the allele, but how many actual individuals. So the genotypes are the phenotypes. For P squared, that's the ones that are homozygous dominant. The 2PQ, and again, if you look, it makes sense because if we take a heterozygous individuals, we have here the P squared would represent how many are homozygous dominant, or in this case, big T, big T. We have 2PQ, so there's two of them that are PQ. That would be big T, little t, and then the Q squared would be the little t, little t. So it basically allows us to predict what or how many offspring there should be, and then if we compare that to what actually happens, if our numbers match, we can say, without question, there is no evolution. So let me throw So here's a sample problem that kind of will work us through real basic, a small population with the Hardy-Weinberg equilibrium, and you can see how it works. But in this case, what I'm looking for is to calculate out the allele frequency, so the P and the Q for this first generation. Then I will plug those into the Hardy-Weinberg equilibrium and say this is how many offspring I should have. And then if I look at it again, I should be able to sort of predict and see, did it actually work in real life? So in this case, the steps are pretty simple. Step one, I always want to do is figure out how many organisms first. So this is really clear. There's two that are homozygous dominant, three that are heterozygous, and two that are homozygous recessive. So 3, 5, 6, 7. Total individuals, I have 7. Now, each of those individuals has 2 alleles for that trait. So to find out the total number of alleles, I just take the total, or the number of individuals, and I take it times 2. So that will give me 14 alleles. All right, so far so good. Now, step 3. For P and Q, all I'm going to figure out is the percentage of, for P, it's the percentage of the dominant allele. For Q, it's the percentage of the recessive. So for the dominant, I look and I say, how many dominants are there? Now, if I go up to the top here, I know that there are 2 that are big B, big B. So there are 2 of those and 2 of those, and then 3 of them that are big B, little b. So if I add those up right there, 3, 4, 5, 6, 7, right? I have 7 dominant. So I take 7 divided by 14, and simple math, I get 0.5 or 50%. Now for Q, common sense would tell me it's got to be 0.5 because these two have to equal 1. But just to make sure the math is correct, I say how many recessives are there? There are, I'll do this in a different color, there are 3 that are little b, and then 2 that are little b. Add them up, I get 7. I divide by the total again, and I get 0.5. So that gives me the allele frequency for those 2 places. Now, again, you might want to calculate it tomorrow if you're not super confident here, but I just plug my P and my Q in. So I plug in 0.5, and I say, what is 0.5 squared? Hopefully you don't have to punch that in, but it is 0.25. Q squared then, I know that one would be also 0.25. And of course, I could do the math. I could plug in 2 times 0.5 times 0.5, but I also know that those have to equal 1. That's what the equation tells me. So I know that this has to be 0.5. So this is, remember, representing that Punnett square, where 0.25% or a quarter will be this one. That makes sense. Another quarter will be this one. And then a quarter of them will be, right, the mix, if you can see my coloration here. So those would be the 2PQ. So it's all based in percentages now. So if I say I'm going to predict how many the F1 will have of each genotype. If I say I know there are 72 offspring, how many of them are going to be homozygous dominant? Well, if I take 72 and I take 25% of it, it gives me a number of 18. Right? I could also do the same. I could say what is half or 0.5 or 50% of 72, so that'll give me 36, and this will give me 18 as well. So that's my prediction. Now, if I went through and I did my math, so there's my population again. Now, if I go through and do the exact same steps that I did up above, if I do these steps, but now I do it for these numbers, I will figure out the new P and the Q. Now, I've already done the math. You can trust me on this or you can double check me. But I went through and I figured out that this case, P is also 0.5 and Q is also 0.5. So I look at this and I say comparison. Is there change or is there no change? Well, in this case, there is no change, which equals no evolution is occurring because none of those conditions were met. So it's a simple way to go through and prove it. Now, if it did change, if the P and the Q did change, then I would say there was change because something was happening. All right, what are those somethings again? Well, that's when we discuss the different reasons why. There were 5 of them, and this is what the Hardy-Weinberg tells us. It could be what we term gene flow. When we have gene flow, right here, gene flow is one of them. That is basically when alleles or individuals move from one population to another. So in these diagrams, this is from your text, but you can see in the upper corner here, there everything is a fourth, right? Blue, white, green, and orange, whatever that means. The initial population has, for some reason, has more green moving in and some orange moving out, so the population now has shifted to this. Now, that subtle shift is going to cause the gene pool to now be different. So the P and the Q for the next generation after this move happens should be different. So differences could be caused by gene flow. That's one option. A second option is mutations. Now, back from our genetic unit, we know that every single person in this room is a genetic mutant. That's a given that we talked about. So mutations would be another reason. In other words, there's a new allele that comes into the situation. In this case, it's a red one. So now from here to here, we now have a little sliver of red. So evolution is occurring. The gene pool is being mixed. That's number 2. Number 3, we could have, or we do have, sexual selection. In other words, again, here, for some reason, the population does this. Why? Well, this is what, with Hardy-Weinberg, for the equation to work, mating has to be random, and we obviously know that that is not the case. Look at all the big showy birds, the peacocks. Why do they have the big feathers? It's to attract a mate. The more successful you are with attracting a mate, the more possibility that you're going to reproduce. That's sexual selection. So in this case, the blue alleles are associated with a blue body color, which is what the mates want. So in this case, the population shifts very clearly towards the blue. Excuse me. That's number 3. Number 4, natural selection also occurs. Natural selection is not necessarily based on the ability to attract a mate, but it's the ability to survive. So in this case, the white alleles, again, from here to here, the white alleles become way more prevalent. Why? Maybe this is an environment where it is snowy, so the white makes them harder to find, harder to capture, harder to be preyed upon. So the white becomes a naturally selected for thing. Therefore, they're going to increase because those are the ones that are going to survive. So natural selection has a role in this as well. And then when you look at the idea, I guess, the big picture in the end, is that in nature, the Hardy-Weinberg equation as a model does not usually work because in nature, those characteristics that we mentioned, they tend to always be there, at least one of them. So there's going to be natural selection. There's going to be sexual selection. Mutations are going to occur. We're going to have this gene flow, this immigration or emigration. And the one I didn't have a slide for is something called, and I'll write it right here, the fifth one is something called genetic drift. Now, genetic drift is a concept that would work in smaller populations, which can happen, island populations, things like that. They also call it the bottleneck effect, where basically if you have a small population and something occurs, and the best example is like a natural disaster. So let's go back to Darwin. We're looking at the Galapagos. We're looking at one island, San Cristobal Island in the Galapagos. Let's say a cyclone comes through and decimates the population on the island. Where there was 1,000 birds, now there's only 100. There's some reason why the ones survived more than likely. They could handle the cyclone better. So the population gene pool is going to be, again, readjusted to sort of show that. That's called genetic drift or the bottleneck effect. So basically, the whole premise of this, guys, is to look at a model, a mathematical model that disproves evolution, but it says that evolution will not occur, but only under these very stringent guidelines. And that in the real world, in nature, populations do have at least one of those 5 things happening almost all the time. Therefore, we expect the populations to actually change over time. We can do this by showing in real data, plugging it into the Hardy-Weinberg equilibrium and seeing if it does in fact work. All right, that's what we're going to be trying tomorrow. Hopefully this cleared some things up. If not, you should very much so check chapter 11 for the evolution unit. 11.4 contains stuff about the Hardy-Weinberg equation, and you can use that as a guide as well. We will try some more problems tomorrow in class, and we will take this one step farther. Enjoy your evening. Go wild.